Myself Sandya R, Associate Professor of Emmanuel Arasal College of Pharmacy, Department of Pharmaceutics. Today we'll discuss about the topic of cultural media and its classifications. What do you mean by cultural media? The food materials or substance required for growing microorganisms in vitro outside the body is called cultural media. The food material or substance required for growing microorganism in vitro outside the body is called cultural media. Then, uses of cultural media. It is important to grow microorganism outside the body for the following purpose. First, to identify the case of infection from the clinical sample so that proper treatment can be given. Second, to study the characteristics of properties of microorganism. Third, to prepare biological product like vaccine, toxins, antigens, etc. Next, composition of cultural media. Composition of cultural media: first, water, energy source, carbon source, nitrogen source, mineral salt, and special growth factors. Then, composition of cultural media: water, energy source, carbon source, nitrogen source, and mineral salt and special growth factors. Then, types of cultural media. Classification based on physical state: solid medium, semi-solid medium, and liquid medium. Solid medium, semi-solid medium, and liquid medium. Classification based on the integrants: simple medium, complex medium, synthetic or defined medium, and special medium. First, two types of cultural media are two types: classification based on physical state and classification based on the ingredients. The classification of based on the physical state three types: solid medium, semi-solid medium, and liquid medium. The classification based on the ingredients four types: simple medium, complex medium, synthetic or defined medium, and special medium. Then first one, classification based on the physical state. Then physical state is solid medium. What do you mean by solid medium? Solid medium is agar is the most commonly used solidifying agent. Agar is the most commonly used solidifying agent. What is agar? Golden and yellow granular powder prepared from seaweeds, not affected by the growth of the bacteria, melts at 98 degrees Celsius set at the 42 degrees Celsius. What is the agar? Golden yellow granular powder prepared from seaweeds, not affected by the growth of the bacteria, melt at 98 degrees Celsius and set at 42 degrees Celsius. That is classification based on the physical state. Classification based on the physical state like solid medium. Solid medium means agar is the most commonly used solidifying agent. Then next is semi-solid media. Such media are soft and are useful in demonstrating bacterial mobility and spreading mobility from non-motility strains. Then pictures available. Visible semi-solid media. Then liquid media are sometimes referred as a broth. Bacteria growth uniformly producing general turbidity. Example, nutrient broth. That is a cover of the first type, classification based on the physical state: solid medium, semi-solid medium, and liquid medium. Then next, classification based on the ingredients. First one, simple media. Example, nutrient broth and agar. Nutrient broth and N-agar. NB constant of peptone, meat extract, and sodium chloride. NB plus 2% of agar, nutrient agar, is equal to nutrient agar. Sample media, nutrient both or nutrient agar, NB constant of peptone, meat extract and sodium chloride. NB plus 2% of agar is equal to nutrient agar. Then is a picture visible in a nutrient agar. Then second one is complex media. First one simple media and second one complex media. What do you mean by complex media? Such as a blood agar. It has ingredients that extract compounds are difficult to estimate. Blood agar pictures. Complex media means such as a blood agar. It has ingredients that extract components are difficult to estimate. Next, synthetic or defined media. Synthetic or defined media, specially prepared medium from pure chemical substance for research purpose and composition of every component is well known. Example, peptone water. 1% of peptone plus 0.5% of NaCl in water. 1% of peptone plus 0.5% of NaCl in water. Especially prepared medium from pure chemical substance for research purpose and composition of every component is well known. Example, peptone water. 1% of peptone plus 0.5% of sodium chloride in the water. Then fourth one, special media. What do you mean by special media? Enriched media, selectively media, differential media, transported media and anaerobic media. Enriched media, selectively media, different media, transported media, anaerobic media. It is a special media. What it means? Enriched media, substance like food, serum, egg are added to the simple media. Second, used to grow bacteria that are extracting in their nutrition needs. Example, blood agar and chocolate agar. The enriched media. Substance like blood, serum, egg are added to the simple media used to grow bacteria that are extracting in the nutritional need. Example, blood agar and chocolate agar. Then picture visible, blood agar and chocolate agar. Blood agar means containing mammillary blood, usually sheep or horse in the blood. Typically at a concentration of 5 to 10% used to isolated organism and defect hemolysis. And chocolate agar means containing red blood cells that have been laced by this thoroughly heating to 18 degrees Celsius, and it is used for growing bacteria such as a Haemophilus infusumia. Blood agar and chocolate agar. Then selectively media. Selectively media, the inhibitory substance is added to solid media to inhibit commercial or contaminating bacteria such as antibiotics, diets, chemicals, alternation of pH, antibiotics, diets, chemicals and alternation of pH. Examples, they are Martin media, selectively for necessary gonorrhea. It usually contains the following combination of antibiotics. Vancomycin, which is able to kill most gram-positive organisms. Colstinin, which has added kill most gram-negative organisms, especially Neisseria. Nystain, which can kill most fungi. Trimethoprim, which inhibited gram-negative organism, especially swarming protozoa. Ozone methylene blue, selectively for gram-negative bacteria. They dine methylene blue in the media and inhibit the growth of gram-positive bacteria. Campylobacter agar. It is used for isolation of Campylobacterial jejuni from fecal or rectal scab. Contain bacteriological charcoal, cephalosporins and amphotericin B. Downstream Johnson's media is a solid media used to mycobacterium tuberculosis. Mycobacterium tuberculosis contain penicillin, altiaceae and linked to inhibit growth of gram-positive and gram-negative bacteria in order to limit growth to mycobacterium species only. That is the classification of the second type. Second type classification based on the ingredients: simple media, complex media, synthetic or different media, and special media. Special media is 5 types: enriched media, selectively media, differential media, transported media, and aborted media. And enriched means substance like food and serum, egg are added to the simple media. Then picture of the blood agar and chocolate agar, selectively media. Examples: camphor agar and nonsense methods. Charcoal containing bacteria charcoals and cephalosporins, instance methylines in the blue. Then methylines in the blue. Thank you.